I am thrilled to be able to present Mycotecture off-planet NIAC Phase 2. Humans need habitats to protect themselves from the elements, and nowhere is this more important than off the cocoon of planet Earth. Now, the traditional method is to bring the habitat with the astronaut, whether they're living in the lunar module or in ISS. Proposals going forward where the habitats are brought with them involve incredibly massive habitats that are just going to bust the upmass budget. So there's got to be a better way. New approaches involve robotic construction of regolith and ice houses, or we have proposed is turning to fungal mycelia. Now, you're all familiar with a mushroom, which is the fruiting body of a fungus. But underneath the ground, you see these fine hair-like structures called mycelia, and these can be used as a building material either by themselves or agglutinating other materials. So this field of building with fungal mycelia is called mycotecture. There are all sorts of advantages for using this on the Earth. It's inexpensive, it's a great material, the flammability is very low, and so on and so forth. Potentially self-healing, sustainable, compostable, green architecture, on-site changes, and so on and so forth. Many of these advantages would also be true using it as a building material off-planet, which is why we've proposed this. And here you can see my major partner in crime, Chris Maurer, who's the principal architect at Redhouse Studios, who has been with me on this the entire way. So what have we done? This is primarily the work of the 2018 Stanford Brown RISD iGEM team, a group of undergraduates that competed in the iGEM, which is a synthetic biology competition. I would encourage you to take a look at their wiki. They were able to show that fungal mycelia could grow on darn near anything that I had in my pantry, but most importantly, that with the addition of nutrients, they can grow on Mars or lunar regolith simulant. They built some little bricks, and here you can see a human-rated stool that they built in about 2 weeks. Some of these materials were then tested at the Soft and Hybrid Materials Facility at Stanford, under the direction of co-I Debbie Sineski. Then there was a project with fungal glues that Kale will explain.
Imagine that you wanted to build a large mycelium structure, so large that you don't want to wait for it to fill out a mold. So you can grow bricks instead, but all bricks need mortar. So what are you going to do? What if I told you you could grow your own mortar too? Welcome to MycoGlue.
In collaboration with the Danish Technical University iGEM team, who also worked on Mycotecture off-planet, and as well as my students and Chris and his Redhouse team, we've done some work looking at protection from UV using melanin and from hard radiation. We've also looked at some enhancements for the fungal mycelia, and this was rather fun. We were able to combine some of what we achieved during a Phase 1 biomining NIAC several years ago, where we developed proteins that could bind copper. And here we then attach the proteins to the fungal mycelia to make a copper filter. This resulted in a PhD thesis, a paper in Scientific Reports, and we have a patent pending. So here are the key knowledge gaps and objectives that were filled in Phase 1. And the one last thing I didn't mention, that under Professor Jim Head at Brown University, there was a Mars architecture that was developed for this. We're now switching over in Phase 2, thanks to the Artemis missions, to focusing on the moon feed forward to Mars. So let me explain the overall concept by showing a video that Chris Maurer has produced for us. So in the near future, a lander delivers a package to either the lunar or to the Martian surface. It would be nice to have it there before the astronauts arrive, because you would like to have your hotel in advance. This package already contains the fungal mycelia, and in the example that he's going to show you, it also includes cyanobacteria. So what happens is this structure is then inflated by the addition of water and gases and grows into a multi-layer building. Well, it inflates into one. Then these cells that are involved, these cellular structures, are filled with the cyanobacterial medium if they are being used. And then they can be used as food material for the fungi. And the result is you end up with a multifunctional composite and the structure that you now have off-planet. So we've started a bit with Objective 1, filling the key technical gaps. I should point out that we were just funded this year, so a lot of my last few months has been spent trying to move money around. Our labs are still not open at NASA Ames, and so much of this work is also being done either remotely or is being done by our fabulous co-Is, of which there is a whole list, as you can see. And so let me tell you what we have done so far. Looking at the mycelia for the strength and the radio protection, we've given some advice to Chris Maurer, who has moved forward, including looking at these very lipid-rich fungal mycelia. You can see a picture that I took over on the right, a micrograph, and that's not a tumor. That orangey blob is a lipid blob that has come out of the fungal mycelium. I mean, these things are just packed with lipids, and we think that they may be providing some resistance from ionizing radiation. There's some testing that Debbie and her students have already done on some of the materials from Chris. And then there are a couple of projects that Chris is heavily involved in that are not being funded by NIAC, but clearly are going to contribute to this overall project, so I listed them there. Very importantly, we now have another member of the team, Dave Cadigan, who is the president of Moonprint Solutions, who has extensive expertise in inflatables, who has been directing us on some of the points that we perhaps missed the first time through on this proposal, including the idea of using drop stitches inside the inflatables. Chris Maurer is already on it, and you can see a little cell that he prototyped. He's filling it up by hand, which is not how we plan to do it off-planet. But I like this video because it shows you the drop stitch construction, and that the result is you end up with a inflatable that is a cube rather than a sphere, which is exactly what you want. So it's very important for maintaining that structure. Upcoming, we'll be able to test some of these materials at a very cool planetary simulator at McMaster University, and we're very grateful, Michael, for being part of the team on that. We'll be able to do both a lunar and a Martian simulation on some of these Mycotecture materials. We'll be able to do other enhancements to the Mycotecture, and this is basically being led by Anil Webber at the University of Newcastle, who in my mind is famous for leading an iGEM team in 2010 that used Bacillus subtilis as a way to repair cracks in concrete. Mission Context is once again under the watchful eye of Professor Jim Head, and he's already been integrating with some of the biweekly architects meetings, his own Friday research meetings, starting to bring students together on this, and has presented an abstract on paper in the future use of Mycotecture for Artemis and beyond at the LIAG meeting September 1st. We've started looking at the benefits and trades, and very grateful for the KSC Swampworks lab for already being in touch with us and meeting with us on in situ construction materials. And so we would very much like to continue this discussion. We do believe that ultimately what will be used off-planet is some combination of Mycotecture and some of these other approaches. So therefore, we've started a bit on the technology roadmap, which will be ongoing. Looking at the impact of this technology for terrestrial materials and applications, there's a huge project that Chris has been leading in Namibia, which is very cool, and I wish I had time to discuss it. We do have one of the top 53 Michelin star chefs, Enrico Atzka, at the Azurmendi restaurant in Spain, who is all about sustainability, and he is very interested to see what we can do for his restaurant in terms of replacing bits and pieces with Mycotecture. We also have Professor Martin Dade Robertson, also an architect who works in Mycotecture at the University of Newcastle and head of the Hub for Biotechnology in the Built Environment, involved in the project. His PhD student, Monika Lipinska, has been involved in this project since she was a master's student at the International Space University in my lab. We've had several outreach opportunities already, several through NASA PAO. These are interviews. Thanks to Mikkel, we've had a very nice feature in the McMaster Deep Science Dive, and there have been several presentations. I gave one at the Mars Innovation Forum. Monica's given several, and we have an upcoming presentation at IAC thanks to Monica's lead, and it looks like I'll actually be there. As I mentioned, we have several papers. We have Jim's from LIAG that was just presented. We have one thanks to Jim's urging at LPSC from last March, and then this IAC one that I've mentioned several times. Thank you.